You now know that cells are the most basic unit of life and that all living things are composed of cells. You also learned that cells can only come from pre-existing cells. In this lecture, you will learn about the various phases of the cell cycle, including cell division, as well as ways that the cell cycle is regulated. We will also learn about what happens when cells lose the ability to control their own division. There are two reasons that cells divide. The first is that the cell becomes too large for its DNA to handle. We call this DNA overload. A cell has a limited amount of DNA in its nucleus, and as it grows larger, there is a greater demand on the DNA to direct cell activities. I like this analogy of a small town library. As the town grows, more and more people borrow books, which can lead to a waiting list or a delay in book borrowing. Think of the cell's DNA as the books and the growing cell as the growing town. The second reason that a cell divides is that the exchange of materials cannot keep up. The ability to transport materials into a cell depends on the surface area or the cell membrane. The need for these materials depends on the volume or the cytoplasm and organelles inside the cell. These don't increase at the same rate. As the cell grows larger, the volume increases faster than the surface area. In other words, food and oxygen cannot get into the cell fast enough, and waste cannot get out of the cell fast enough. When this happens, the cell needs to divide. Before we get into the various phases of the cell cycle, we first need to understand DNA. All cells contain genetic information in the form of DNA. Prokaryotes, like bacteria, have a single strand of DNA that is circular and floats freely around the cytoplasm of the unicellular organism. It is not contained in a nucleus like eukaryotic DNA is. In non-dividing eukaryotic cells like plants and animals, the nuclear DNA is spread out into a stringy-looking substance called chromatin. In cells that are dividing, this chromatin becomes more organized and condenses into structures called chromosomes. Chromosomes are rod-shaped structures that resemble the letter X during cell division. In preparation for division, they make a copy of themselves called a sister chromatid. This copy is identical so that the DNA in the cells resulting from division are identical to each other and to the original cell. The two chromatids, the original chromosome and the copy, are held together by a centromere. The name of the centromere is easy to remember because it holds the chromatids together at the center. Chromosomes come in pairs because they are inherited during reproduction. Humans have 23 pairs of chromosomes, making 46 total individual chromosomes. During reproduction, each parent makes a copy of half of their own genetic information to give to their offspring. As a result, the offspring inherits matching sets of chromosomes, one from mom and one from dad. The matching sets are called homologous chromosomes. Homologous chromosomes are the same size, same shape, and carry genes for the same traits, but they are not identical. The reason they're not identical is because they don't have the same choices for each trait. Remember, one is inherited from mom and one is inherited from dad. For instance, if eye color was controlled by a single gene, you may inherit a gene for blue eyes from your mom and a gene for brown eyes from your dad. These genes are located in the same position on homologous chromosomes, but they have different choices. This diagram is a visual representation of the cell cycle. The four phases are G1, S, G2, and M. In G1, or growth 1, the individual cell is growing larger. In S, or synthesis, the cell makes a copy of its DNA in preparation for division. In G2, or growth 2, the cell is growing and doing more work to prepare for cell division. Then, in M phase, or mitosis, the cell actively divides into two new cells. Together, G1, S, and G2 are known as interphase. Think of interphase as the in-between phase because it is the phase that a cell is in when it is not actively dividing. The cell spends most of its life in interphase, specifically in the G1 phase, where it is just existing as a normal cell and carrying out its specific function. When observing a cell in interphase, you will be able to see the nucleus containing chromatin. The M phase, called mitosis, is a short phase where the cell actively divides. The cell first goes through a series of stages where the nucleus divides, followed by the division of the cytoplasm. Division of the cytoplasm is called cytokinesis, and it marks the end of the cell cycle. The first phase of mitosis is called prophase. A good way to remember that prophase comes first is to remember that pro means first. During prophase, the DNA condenses into chromosomes. Cell structures known as centrioles and spindle fibers appear. Their function is to attach the centromeres and organize the DNA during division. The cell membranes surrounding the nucleus and the nucleolus disappear during this phase. The second phase of mitosis is called metaphase. During metaphase, the chromosomes line up along the equator in the middle of the cell. The centrioles have migrated to opposite poles and the attached spindle fibers line up the chromosomes. This is one of the easiest stages to identify when observing cells under a microscope because the chromosomes are lined up in the middle. Remember, metaphase and middle. The next phase of mitosis is called anaphase. During anaphase, the centromeres split and the spindle fibers pull the matching sister chromatids apart in order to create two new cells with genetically identical information. You can recall what happens during anaphase by remembering that anaphase and apart both start with the letter A. The next phase of mitosis is called telophase. Think of telophase as the opposite of prophase. The chromosomes now spread out into chromatin and two nuclear membranes form. The centrioles and spindle fibers dissolve because their job is done. The nucleus has been divided and mitosis is almost complete. The final step is the division of the cytoplasm, also known as cytokinesis. Sometimes cytokinesis is considered a part of telophase. Cytokinesis looks different in animal cells than it does in plant cells. In animal cells, the cytoplasm pinches into two new cells, and this is identifiable as a cleavage furrow. In plant cells, the rigid cell wall is unable to divide this way. Plant cells divide by growing a cell plate down the middle. This cell plate eventually turns into the cell wall and the two new plant cells. Now that you have learned about the process of cell division and the various phases of the cell cycle, it is time to talk about how the cell cycle is regulated. Cells have specific genes that regulate division. When needed, they turn on to stimulate cell division. For instance, when you cut your finger, the skin cells near your cut are stimulated to divide in order to repair the damage and heal the wound. Cells divide at different rates. Some cells, like hair and skin cells, divide frequently, while other cells divide slowly, and some just don't divide at all, like nerve and muscle cells. There are two different ways that the cell cycle is regulated. This is first by internal factors, or proteins that respond to activities inside of the cell. For instance, cyclin is a protein that regulates the timing of the cell cycle and causes spindle fibers to begin forming. There are also checkpoints built into the cell cycle that prevent the cell from continuing if it is damaged. In G1, at the checkpoint, we're asking if the cell has doubled in size. In G2, we're asking if the chromosomes have been copied. And in M, we want to know if all sister chromatids have attached to the spindle fiber before anaphase. These are all things that the cell is asking about itself or looking into before moving on to the next step. There are also external factors that regulate the cell cycle. These are proteins that respond to events outside of the cell. Growth factors activated by neighboring cells stimulate cells to divide. This is especially important during fetal development. Another external factor called density-dependent inhibition sends a signal for cell division to end when cells touch each other. This prevents excessive growth. Normal cells continue to grow until they contact other cells. When cells lose their ability to control division, they become cancerous. They do not stop when they come into contact with other cells and just keep dividing. This is what makes a tumor. There are two types of tumors. A benign tumor is not cancerous. It is just an abnormal growth. Benign tumors do not spread to other parts of the body and can easily be removed. However, benign tumors can become cancerous. Malignant tumors have spread to other parts of the body. This is known as metastasizing. Part of the tumor breaks off and enters the bloodstream, which allows it to travel to other areas and spread the cancer. Cancer can be caused by many different factors, but they all affect cells in the same way. They cause a mutation in genes that control cell division. Some cancer-causing mutagens include tobacco and radiation. Cancerous mutagens can also be inherited during reproduction. Normal cells have a form of programmed cell death called apoptosis. It is your body's way of eliminating cells that are imperfect. For example, a precancerous cell or a virus-infected cell. However, cancer cells are able to escape apoptosis and divide uncontrollably. While we haven't found a cure for cancer yet, there are many treatments available. The most common treatments are chemotherapy, which is a group of drugs used to target and kill cancer cells. Radiation, which uses high energy wavelengths to target and kill cancer cells, and surgery, in which isolated tumors are removed from the body. The problem with treatments that target cancer cells, like chemotherapy and radiation, is that they target all fast-dividing cells, and they cause harsh side effects for the patient. A doctor and patient will choose the treatment plan that works best for them depending on the type of cancer and how far it has progressed. Some cancers, like leukemia, are not able to be surgically removed. Some newer methods of cancer treatment include stem cell transplants, immunotherapy, and hypothermia treatment. Many patients choose a combination of treatments to increase their chance of survival. We've now finished our notes for cell cycle and division. You want to make sure you have everything before moving on to the next assignments, and review the answers to your questions before completing this video. If you have any questions, please let your teacher know.